we will discuss the replisome assembly. This is part 1 of the replisome assembly, which builds on the origin unwinding video. I hope you have watched that already. I can probably make a much quicker video explaining the replisome, but I think there are two solid issues that are often overlooked by most people when understanding the fundamentals of DNA replication initiation. The first issue is that DnaC protein, which loads helicase, also inhibits its activity. The second issue relates to the fundamental observation that primase and helicase complexes physically bind to each other, but their movement is incompatible with each other. So we will talk about replisome assembly by looking at DnaC, DnaB, which is the helicase, and DnaG, which is the primase. And I hope you will appreciate the process a bit better and see how beautifully these proteins interact with each other. Let me start by drawing out the unbound origin, which is the place where we left off in the origin unwinding video. And let's get back to these DiaA proteins that we said act like molecular glues to aggregate the DnaA proteins on the origin. Now, following this origin unwinding, the DiaA is released, or rather, it has to release. And although the exact mechanism for its release is unclear, people have speculated that there might be a DiaA release factor, but it is generally not known. Sometimes, this job of DiaA is also done by another set of proteins called Hu proteins. But the point remains that DiaA has to be released after the origin opening. Now, you may ask, why do DiaA protein needs to dissociate away? After all, it stabilizes the DnaA on the origin. The answer comes from the fact that the binding of DiaA on the DnaA protein occurs on domain 1, specifically at phenylalanine at position 46. Now, the transition into the replisome assembly, which starts by helicase loading, actually depends on whether or not this phenylalanine 46 is available. So, it needs to be accessible. But if DiaA is bound to that amino acid, it is not. And therefore, DiaA needs to be released from the origin, or at least from the DU region. Now we can get into the crux of this video. And let's start by discussing helicase loading. Helicase, also known as DnaB in E. coli, its loading depends on a loader protein called DnaC. This idea is similar to the loading of RNA polymerase at the promoter, which in prokaryotes depends on sigma factor, and TF2F in case of eukaryotes. Some people actually say that DnaC is not actually a loader protein, given that it never actually loads the helicase. It just increases the frequency of loading of the helicase at the origin. But for the sake of simplicity, we will stay away from the semantics and call DnaC a loader protein. The active form of a helicase is a hexamer, meaning that it is formed by 6 monomers, and the whole complex is stabilized by magnesium ions. A monomer of the helicase may look something like this. It has an N-terminal domain and a C-terminal domain, and 6 of these monomers form an active hexamer. So you have this back end, N-terminal, the front end as the C-terminal. More specifically, the N-terminal is the smaller end, and the C-terminal is the bigger end. This hexamer is like a cylinder, and the internal channel is positively charged, which is convenient because DNA is negatively charged, so it has some nice ionic interactions. Now, one very, very important fact to keep in mind is that active helicase moves in the 5' to 3' direction on the template DNA. The larger C-terminal domain is the one which has the helicase activity, and it also has the ATPase subunit to power the helicase forward. Okay, now to understand the loading of the helicase, we have to understand the loader protein DnaC. To load the helicase, the DnaC protein interacts with the domain 1 of DnaA proteins, which are already at the origin, and the loader protein also interacts with its domain 3. The additional help in the loading of helicase comes from the helicase itself. The N-terminal domain of the helicase interacts with this phenylalanine 46 in the DnaA domain 1. This is the same amino acid at which the DiaA was bound. Now, let's try to visualize the process of helicase loading. To add some details about the loader protein, you should note that DnaC binds to the C-terminal end of the helicase, and usually 3 of them bind to the hexamer. Now, this DnaC protein interacts with the DnaA domain 1 and 3, and loads the helicase at the DU region, where the DnaC is bound to the C-terminal end of the helicase. Notice the positioning of N-terminal end and C-terminal end of the helicase. The strand is 5' to 3'. The 5' side has the N-terminal region, whereas the C-terminal region is facing towards the 3' end. And as we have said before, typically you will find that there are 3 loader proteins present on a hexamer. So the ratio of molecules in loader to helicase is 3 to 6. The 6 comes from the fact that helicase is a hexamer. Now let's complete this picture of helicase loading. Note that this top strand goes from 5' to 3', and then there is another 5' to 3' strand in the anti-parallel direction. And given the loading direction of helicase, we can draw out the helicase on this bottom strand as well. The N-terminal domain is at the 5' side, and the C-terminal domain is on the 3' side. So essentially this means that the loading of helicase on this top strand is mediated primarily by DnaC and DnaA interactions, whereas the loading on the bottom strand is mediated primarily by the N-terminal domain interacting with the domain 1 of DnaA, and specifically by the phenylalanine interaction. This doesn't mean that the N-terminal domain of the other helicase does not interact with DnaA, but its position suggests that that interaction in the other case is less frequent. And likewise, the DnaC position on this helicase at the bottom strand means that its interaction with DnaA is less frequent. So to sum up, we have the DnaB N-terminal interface and the DnaC interface that mediates the helicase loading. To understand the rest, we must look at some aspects of the helicase structure. This diagram here is a side view of the structure. Now I want you to consider the N-terminal and the C-terminal views, which is basically the top and bottom view. The C-terminal view is straightforward. It contains a fairly homogeneous hexameric organization that looks like a cylinder, and it has a central channel which is open. The N-terminal view, on the other hand, reveals a very different story. It has 3 structures that look like dimers that come together as a trimer. And each of these dimers has this notch kind of structure. We will see in a moment why this organization is important. Now, to move to the next step, I will simplify things and only use the top strand for explanation. So far, we have a DnaC and a DnaB organized on the origin, where DNA passes through the positively charged channel of the helicase. To recap an important point, the helicase moves in the 5' to 3' direction, and its helicase activity comes from the C-terminal domain. However, currently the DnaC protein has occupied the C-terminal domain, and this essentially blocks the helicase and ATPase activity of the DnaB hexamer. This brings us to the first issue that I want to go over. The loader protein of the helicase is also its negative regulator. So if helicase has to move ahead, the loader protein must be released. And this release occurs due to the recruitment of DnaG, which is the primase protein that synthesizes a primer. Now, I could have stopped here with my explanation, but I want you to appreciate the elegant mechanism by which the recruitment of DnaG kicks out this loader protein DnaC. To help you understand the mechanism, you should note that the DnaG loads at the N-terminal domain of the DnaB hexamer. For your reference, the N-terminal domain is this other end of the helicase, which is free. DnaC is bound to the C-terminal end. Simply speaking, the binding of DnaG at the N-terminal region causes a conformational change in the hexamer. And this conformational change activates the ATPase in the DnaC. And the ATP hydrolysis causes the loader protein to be released from the helicase. But how is this conformational change occurring? And this is where the N-terminal view comes in handy. Remember I said that these notches between dimers are special? Well, it turns out that DnaG binds at these notches in the N-terminal domain of the helicase. And this is what it means when we say that DnaG is loaded onto the helicase. And this binding of primase at the N-terminal region locks the helicase position so that it can engage the DNA in the central channel much better. This locking causes the loader protein to be released. Now, interestingly, here also there are 3 DnaG proteins for the entire helicase. This is the same ratio as we saw with the DnaC. Now, let's complete the structure of the helicase with the primase bound to it. In this view, the DnaC is released and the primase is bound to the N-terminal region, and this primase makes the primer. This now brings us to the next problem, which is the incompatibility in the primase and helicase movement. To understand that, you have to know that DnaG is basically a specialized RNA polymerase. It uses DNA and makes RNA. Now, let's define the issue of incompatibility with the helicase. As we have noted so far, the helicase moves from 5' to 3' direction. Now, DnaG makes RNA, and its synthesis direction is also 5' to 3', which means its movement on the template must be from 3' to 5'. But the movement of helicase on the template is 5' to 3'. So helicase wants to move forward, but the primase, which is physically connected to the helicase, wants to move backwards. Hence the problem, the tug of war, the DnaG and DnaB, which are active in the opposing directions. So how does this actually work? How is it not a problem for the cell? To understand this, I will focus on the side view of the helicase. And for simplicity, I will only talk about this dimer here. From the side, this will look something like this, where DnaG is connected to the N-terminal domain of the DnaB, and the single-stranded DNA runs through the channel of the helicase. The helicase is moving towards the 3' end. The active site of the primase, which contains the RNA polymerase domain, is at the distal end. And before the DNA gets to the RNA polymerase site, the single-strand DNA is looped into it. I won't go into the details of how it gets looped, because that can be a video in itself. Now, this active site tries to recognize the priming site, which is specifically CTG on the single-stranded DNA. Let's zoom into this to get a better idea of what I mean. So the recognition of the priming start site occurs when a CTG arrives in the active site. So that is a high-affinity recognition site for the primase to start making the primer. And the first nucleotide to be synthesized is the adenine and then guanine, and it moves to the 3' end. So this G here is only used for recognition and not as template. Okay, now back to the problem. The primase wants to synthesize the primer towards the 5' direction on the template, but the helicase is moving towards the opposite direction. And here comes the solution. What actually occurs here is that after the CTG recognition, the DNA is not allowed to pass through the primase, but instead the synthesis begins on the template. Since helicase keeps moving ahead, a loop of single-stranded DNA comes out in between the primase and the helicase, and the primer synthesis can still occur. Now just keep in mind that the primer will have uracils in it, given that it is an RNA polymerase. This short loop is stabilized by some single-strand binding proteins. So this is how recognition priming site by DnaG and helicase movement are coordinated. This also means that the RNA are relatively short. They range from 10 to 60 nucleotides, but 90% of the time, they're only about 11 or 12 bases in length. If you have long primers, then the size of the single-stranded DNA loop between the primase and the helicase will get too big, and that will destabilize the structure. And this is a good transition to the next step. So we have overcome the compatibility issue, but why are these primers so short? What determines the length of these primers? To answer that, we have to look at the full structure of helicase complex. So far, I've simplified the outlook to a single primase, But remember that in a full hexamer, there are 3 primase bound to the N-terminal region. And out of the 3, only 1 primase is actually functioning to make the primer. And I'm going to call it the active DnaG. And here is the loop that comes out between this active primase and the helicase. So, one control of the primer length comes from the fact that the flexible loop cannot get too long. If it does, then the loop and the corresponding RNA-DNA duplex will destabilize the active primase and cause its release. The other mechanism relies on the other primases, which are not active. These primases are also pretty flexible. And there are some studies that have shown that the mere dangling of these non-active primases will interfere with the activity of the active primase, and that'll cause the active primase to release. And this also appears to be the reason for control of the primer length. There's actually one interesting fact that relates to the DNA polymerase activity in bacteria, which fits very nicely with the short primer length. If you know how RNA primer length and DNA polymerase activity in the replication are related, comment down what you think that relationship is. My answer to this question will be in the next video. Anyways, back to the primase and helicase complex. Now, after regulation, the active primase is released, and sometimes all primases are released from the helicase. But in most cases, 1 or 2 primases stay bound. Now, in this complex, we have 2 primases, and the RNA primer, which is left behind. Oh, and another fact. The length of the DNA in this helicase is around 20 nucleotides. Okay, here's one last thing before we start to wrap up. When the primase is actively making RNA and the helicase is moving, the DnaB movement is actually pretty slow. But when the active primase is released, the helicase becomes faster. This is not surprising to imagine because of the intermediate single-stranded DNA loop that appears between the primase and the helicase. This also shows that primase and helicases are indeed a bit incompatible when it comes to the optimal speed of the helicase movement. Excellent. I hope this helps you understand some key ideas. Let's bring all this information in a grand picture of the origin opening. Now we have the DnaG and DnaB complex on one strand, positioned accordingly, and this position of the top complex occurs at the L region of the DU element. And the positioning of the bottom complex occurs at the R region of the DU, which partially overlaps with the DNA trios. This is the activated state of the helicase, where an RNA primer has been made. Now, the helicase can actually move and unbind the genome starting at this origin. The actual unbinding process will be discussed in the next video, which will also cover the loading of DNA polymerase to form the complete replisome.